Welcome to this episode. I'm your host, Arshad Khanani. And for this episode, I'm joined by my good friend and world-renowned retina specialist, Dr. Dante Pieramici, director at the California Retina Research Foundation and partner at California Retina Consultants in beautiful Santa Barbara. Hi, Dante.
Hey Arshad, been looking forward to this conversation with you.
Well, I'm thrilled to have you here. And, being an expert in gene therapy, you have been involved with so many different programs. So, in this episode, we'll discuss gene therapy in the treatment of retinal diseases, as well as the recent and the latest data from clinical trials. So in this podcast, Dante, before we get into discussion about the design and data in gene therapy, we ask our faculty a few questions. So tell us, how did you end up in retina?
That story is so long ago now. I have to really look in the back of my memory to find that. But I was in Baltimore in medical school some years ago, and I was a neuroscience major. So going into medical school, I really was kind of an open book. I didn't have any preconceived ideas of what I would do, but I thought I'd probably do something in the neuroscience field. So I did some neurology, neurosurgery rotations, and I think it was one of the neurosurgeons. It wasn't Ben Carson, but he was there separating twins at the time. But it was one of the neurosurgeons who suggested that I should do a rotation in neuro-ophthalmology with Neil Miller because it would be a great training to be a neurosurgery resident or fellow in the future. So I went to Neil Miller, who was at the Wilmer Eye Institute at that time, and he said, well, I want you to do some basic ophthalmology stuff. So I worked with Neil and Susan Bressler for a month first, and then I worked with Neil Miller. And after that, I was really more interested in ophthalmology, and particularly retina. I just liked the technology. The patients weren't, where it was a more positive field, I thought, particularly at the time. There was just many more things we could do, but it was also an innovative field. I think there was a lot of things that hadn't been solved, in particularly in retina, and so I went to Emory then and worked with Paul Sternberg and Tom Alberg and Tony Capone and Dave Saperstein and the group down there, and they really got me interested in the surgical aspects of retina as well. So, yeah, that's how I got involved in it. And I think I've heard that story before, that other people through the neurosurgery route, they went into retina. And I guess we are neurosurgeons, just of the brain that's outside of the skull, right? The part of the brain.
No, this is inspiring. I mean, you trained with some of the legends in the field, and I'm so glad that you didn't choose neurosurgery because you have contributed a lot to our space. So that's great, Dante. And now some fun stuff. Tell us about your favorite hobby other than having long hair and putting a lot of gel and product in it.
Well, I mean, I guess my favorite hobby is probably traveling with my family and experiencing new things around the world. We're a pretty active family, so when we travel, we like to bike and hike and kayak and do all of these things. So that's probably my favorite hobby. Although I do a lot of cycling here in Santa Barbara and some in Utah. I like to road cycle. It's a dangerous hobby, like my other hobby, which is downhill skiing. So yeah, I just don't have a good sense of picking a safe sport. But yeah, those are the two things I like to do, but mostly to do them with my family in the free time.
No, that's great. You're very active. You've been in the field for a long time. You're forever young. And then the last one, what's your favorite vacation spot?
I guess my favorite vacation spot is the spot I haven't gone to yet. So, it's always exciting to find new places. And as you know, we travel a lot for work, and we go to different places around the world for the different meetings and things. And those are fun. I mean, it's fun to see colleagues, and I really missed that the last couple of years with COVID. But getting together, that's always fun. But traveling again with the family and finding new places to go. I mean, if I had to pick a place, it'd probably be Italy, and I don't know why that is, but I think I just love Italy. For the food and for the scenery. And I guess I have a little heritage that dates back. So it's somewhere in my DNA or my, epigenetics. There's a desire for the motherland. So it's always a favorite place to go back to.
Oh yeah, you can't go wrong with Italy for sure. So no, that's a great spot. So let's dive into our topic at hand today, Dante. let's talk a little bit about gene therapy. And looking at gene therapy, can you first explain gene therapy for inherited retinal diseases versus for common retinal diseases for our audience?
Sure can. I mean, when you have an inherited retinal disease, you've got a genetic abnormality. I mean, for recessive diseases, it tends to be that they're not making the proper protein. And for an inherited retinal disease, you can imagine actually supplementing or putting in a gene or a piece of a DNA that may cause the cell now to start making the protein that's missing. And we see this, like, for instance, with Leber's congenital amaurosis. We supplement the missing protein, and we can actually help the patients. And that's kind of a no-brainer, right? You have a genetic abnormality, you're not making the right protein. You use the technology of DNA or RNA to have the cell produce a protein that you need. Now, for some of the autosomal dominant diseases, oftentimes you're producing an abnormal protein that could be in the way toxic. So for these diseases, I think we're going to have to look to things like gene editing, using things like CRISPR-Cas9 to try to fix the DNA. So it's not making the abnormal toxic protein, but now starts making the normal protein for cells. And there's lots of work in our field being done on inherited retinal diseases. And, it's really, we all know about voretigene nevirovec, for instance, being used and commercialized now for patients with Leber's congenital amaurosis. But there's a lot of work being done in other diseases such as choroideremia, X-linked retinoschisis, X-linked retinitis pigmentosa. And I do see a future where, you'll see a patient with what you think is an inherited retinal disease, and then you'll do a DNA sequence. You'll figure out what's wrong in the DNA, and you'll be able to then customize treatment for that patient to either supplant a gene that's not making the protein you need, or to correct the gene that's making an abnormal protein in our patients. So, yeah, I think for an inherited retinal diseases, that's what's going on. There's lots of work going on for that. And I remember when I started in retina, there were people who specialized in genetics of retina, and they were basically just classifying things. And it was sort of sad because there wasn't much you could do. But now this opens up a whole new possibility for treatment, for better vision for these patients. And, as I see these patients in my clinic, particularly the young ones, I know that their future is going to be very different than the people that had it 30, 40, 50 years ago who have lost a lot of vision and things. So that's for inherited retinal diseases. I guess the only other thing I'd add to that, too, is that there's optogenetics, which is a way that we may be able to use parts of the retina that are still intact in patients that have retinal degenerations and turn them more into photoreceptors. So it's a really neat application of gene therapy that may be usable in many different types of retinal degeneration. So that's inherited retinal diseases. Now, in more complex retinal diseases such as macular degeneration or diabetic retinopathy, there may be a role for gene therapy as well. But in this case, it's going to be using gene therapy as a biofactory. If we want a molecule, say like a VEGF suppressor, like ranibizumab, instead of giving an injection every month, we'll now give an injection or do a surgical procedure to deliver genes to the cells now that are going to cause the cells to use the mechanisms of the cell to produce a protein, the VEGF suppressor. And so this is a biofactory. So instead of correcting a genetic abnormality, we're actually using the cells to deliver drugs. And, in my mind, I think 10, 20 years from now, it won't just be in ophthalmology that we'll be doing this, but, for chronic diseases where patients need to take medicines, for a long period of time or for a lifetime, this may be a way of improving the patient's use of the medication, improving the effectiveness of medications in a real world setting. And I think that this is really exciting application of gene therapy. I think in our field, it has applications to disease processes that we're doing frequent injections like anti-VEGF suppression in age-related macular degeneration, perhaps complement suppression. This would be a great way to produce these complement inhibitors locally and have them be perhaps a one and done therapy for some of these patients. So I think this offers us a different way of delivering drugs to patients that may improve the outcomes in the long run with a steady delivery of the drug rather than this pulsatile delivery that sometimes patients miss appointments and they miss treatments and their visual outcomes go down slowly over time, as we've seen in real world data.
No, I think those are great points, Dante. I think for IRDs, there's so much work being done. And, of course, the bar is very high. It's not easy to get there. But I think as a field, we continue to look for treatment for those patients with inherited retinal disease. And then for biofactory approach, right? I mean, I think, as you said, we have great agents currently, but the outcomes are terrible in the real world for a good number of patients because of the treatment burden issue. Even though we have longer acting drugs that can go, a few weeks more than the approved drugs in the past, but I think there's still a big need. And of course, we have the tyrosine kinase inhibitors and the port delivery system that you and I have involved with closely, all those modalities. But I think gene therapy is different, right? Because it's a one-time treatment. It's kind of like essentially coming to a cure in a subset of patients. We, of course, thought that it's going to be cure for majority of the patients, but we know that each patient has a different need. And I think even reducing burden, I think it's an important aspect that we can do with gene therapy if you're not able to completely cure the disease. So Dante, you are involved with intravitreal, suprachoroidal, and subretinal trials of gene therapy, and we'll go into each one of them. But just tell us a little bit about delivery approaches and the risk and benefits of them.
Yeah, certainly in our space, we're sort of privileged from the standpoint that we have different ways that we can deliver these drugs locally to the eye without having to expose the patient to systemic administration of gene therapy. Because I think people would be very worried about that, right? Because some of these blocking agents are things we wouldn't want to block throughout the human body or alter throughout the human body. So right now there's really three approaches to delivering drugs to the retina. I think the oldest one is really using a vitrectomy approach and then delivering the drugs in a subretinal location. And this makes a lot of sense, I think, if you really need to get the drug to a specific area, right? You can do a bleb and place the drug there. It's a relatively straightforward procedure, in my opinion. I mean, we're all comfortable doing a vitrectomy, and this is essentially making a small bleb under the retina. So pretty straightforward, but it is a surgical procedure and there are surgical risks. And I think about AMD patients, some of them are older and really don't want to go to the operating room and such. So there's some drawbacks to that, I think. And it's a privileged space, the subretinal space. And so we've seen less incidence of intraocular inflammation, either in the vitreous or the anterior chamber. However, there are some changes we've seen with some of these patients, and we've seen this with the patients, the inherited retinal disease patients. There are some pigmentary changes that can occur. And the reason for this loss of pigmentary cells is not exactly clear. It may be an inflammatory reaction, and it very well probably is, or it could be that somehow we're altering the cells by making them biofactories now, and particularly where the drug is very concentrated. And, we may be losing these cells or altering the cells or altering the pigmentation in the cell. So, yeah, there's a lot to learn about that. So that's number one, a vitrectomy approach with subretinal delivery. There's also another approach, the GT005 used a subretinal approach as well, but they're using this orbit system. It's a way of using sort of a suprachoroidal delivery that's very targeted, and then they go into the subretinal space. I think it's, in my opinion, it's relatively complicated for what needs to be done, at least for the biofactory approach. I think it's a great tool for delivery of cell therapy. If you want to get cells in a location, I think it's very important that they don't go into the vitreous cavity because it can cause PVR and proliferation and such. So that's another surgical approach. There's the intravitreal injection approach, and I think this is probably the one we're most familiar with. And this is just using a gene therapy such as ADVM-022 has taken this approach where we're just injecting the gene therapy right into the vitreous cavity, and then it sort of percolates around and transduces cells that start producing the protein. And they could be very distant from the area of disease, for instance. It's not necessary that the cells producing this are right in the macula, for instance. And in fact, we we don't even necessarily need that to be. We just need some cells to be producing the anti-VEGF agent. But the problem with this approach, it being easy, is the good thing. The bad thing is it seems like we have more cases of inflammation with this, intraocular inflammation, and meaning that you're going to have to prophylax with steroids and maybe oral steroids, and there's a drawback to that as well in patients. So we've seen this with the number of trials. The PRISM trial, 4DMT is an intravitreal approach, as well as ADVM-022. And so I think those are two that are going with that. Okay, and then there's the suprachoroidal approach, and this is, been done using the microneedle, and we've done this with steroids in the past and other agents, but now using gene therapy. S1E4 has done this approach for both wet AMD in their AVA trial, as well as diabetic retinopathy in the Altitude trial. And this may be the best of both worlds. You may get a privileged space that, doesn't get a lot of systemic absorption. There may be lower inflammatory responses, although there doesn't seem to be no inflammatory responses. We are seeing some episcleritis and some intraocular inflammation in some of these cases. But it's an in-office procedure, not a surgical procedure. And the patients tolerate it pretty well. It's a little harder than an intravitreal injection, in my opinion, but I think you can become pretty comfortable with this. These microneedles work fairly well. And it may be the kind of best of both worlds approach. So we have the subretinal approach with a vitrectomy or from the suprachoroidal orbit device. You have the intravitreal injection approach, and then the suprachoroidal approach. And these are all being investigated.
No, Dante, that's an excellent summary. So, I think we are most ahead with the subretinal approach, as you said, with our GX314. That is in pivotal atmosphere and ascent studies. And of course, they're ongoing. But based on, your experience and my experience, I think we saw pretty good durability of treatment and protein production in the phase 1b/2a program that both of us participated in. And then, of course, the intravitreal approach being easy. But I think as a field, I feel like, and tell me if you agree, we're learning that dose matters in terms of how much dose or vector genomes you're putting in the eye. I think we need to lower the dose so that we have better safety, especially when it comes to intravitreal delivery. We also need to have better prophylaxis up front so that we don't have indolent, chronic inflammation or long-term inflammation. And I think we need to have vectors that are actually prone to transfect the retina and the choroid instead of the front of the eye, because we did have some serious adverse events in the DME study, the Infinity trial with ADVM-022. So I think those are the learnings that are being taken forward. With ADVM-022, we have the Luna study where there's better prophylaxis and lower doses of ADVM-022 being used. 4D program, started with lower doses anyways and went even lower. And then, you mentioned GA and the GT005. Obviously, gene therapy would be great for patients with geographic atrophy so that they don't have to have a significant burden of injections for the rest of their lives. But of course, the bar is very high in terms of hitting the efficacy we need. So, of course, those trials are also ongoing, and we're waiting for the control data. And then the last one, of course, J&J 1887 is also an intravitreal program. So what are your thoughts on the dosing and the prophylaxis? I think for intravitreal gene therapy and even suprachoroidal, right, we added prophylaxis because there was some inflammation, even though mild and reversible, without prophylaxis. So what are your thoughts on those two things?
Yeah, I mean, you brought up a lot there, Arshad, and a lot of good information. And I do think that the dose means a lot. I mean, the lower dose of vectors, and I think it's probably the vector more than the protein that's causing the inflammation. These adeno-associated viral vectors are giving us more of the problem with the inflammation. So the lower the dose you can go, the better. And again, the ADVM-022 was when they were in the 6E11 type range is where they had more inflammation, and now we're cutting it back to 2E11 and even 6E10 type dosage and finding much fewer cases of inflammation with an individual injection. So yeah, dose matters. I think you mentioned 4DMT, the PRISM trial, they're looking at actually much even lower doses. And this may be for two reasons. One, they have a viral vector that's been evolved so that it's more selective to getting perhaps to the cells we need. So lower dosages of it may be effective. And it's a dual transgene. So they have two things going on. They have an anti-VEGF molecule and a small inhibiting RNA to VEGF-C. So this dual therapy may allow for lower viral vector dosages. And this may be very important to having a low inflammation. So yeah, I think dosages matter. When you get into the suprachoroidal space, you end up having to use a little bit higher doses, I think, to actually get the transduction and to get it to where it needs to be in these cases. And I think, again, we're trying to figure out the right dosages in the different locations. But I think prophylaxis is very important as well. I think that if what we want to do is stop the inflammation from ever occurring if we can. So putting patients either on topical or perhaps intravitreal injection of a steroid implant like a dexamethasone implant, for instance, or oral steroids. These are all approaches, or a combination of these approaches, that are being looked at really for the first weeks to months after the gene therapy to reduce the chance of these inflammatory reactions occurring. And a lot of them don't seem to be all that associated with the neutralizing antibody levels either, at least for the inflammation. So it's hard to predict necessarily which patient may have an inflammatory response up front, a priori to the injection. So I think that these are important things. Trying to find the lowest effective dose, number one, will reduce your inflammation. And then number two, trying to find an effective prophylaxis so that we don't have to run into these problems. I think, with all these gene therapies, our uveitis colleagues are going to be busy in the future managing some of these.
Well, I hope not, right? No, I agree. I think it's been a learning experience, at least for me, being involved with all the programs, just like you are. And I think we are making headway. So I think the inflammation profile in 4150 program looks excellent in that sense that we don't have really any patient with significant inflammation with long-term data that was recently presented at ASRS. And even with the low dose of ADVM022, we saw that the inflammation was much less. So I think as a field, we are learning and we are moving towards the future. And I think, I'm really excited about gene therapy 5 or 10 years from now, where at least we may be able to tell our patients that, hey, if you get this one treatment, there's a good chance, whether it's 50%, 60%, that you would never have to get any more treatment, or we are significantly reducing burden for the others. So no, it's very exciting, Dante, and thanks for being here and for your expertise. Two other questions we ask to all our faculty before we sign off is, what is the career achievement that you are most proud of in your career, Dante?
Well, I have to say, probably, helping build our practice, the California Retina Consultants. I mean, I joined two great guys, Man Nasser and Bob Avery, and they had a fairly good practice going. But, working with these guys, Bob really had the idea that we could be a clinical practice and do clinical research. And, Arshad, you've developed the same model in your practice, that I think in a private practice setting, that we can do excellent clinical research. And actually, we do more clinical research now in the private practice setting than the university setting. And I think it offers our patients the ability to be part of things early on. And we both have these patients now who've been getting intravitreal injections for 15 years. And some of them got it in the clinical trials early on and are still seeing and driving 15 years later because they were part of these trials and they would have lost their vision. So I think there's a lot of value. And we built this practice over the years. And I think that's what I'll be most proud of. All the patients we cared for in the practice and our staff. We've developed an excellent staff over the years. And I'll be proud of what we've accomplished as a group, the team of doctors, the team of our clinicians, our research personnel, the administrative faculty that we have. I think it's been pretty amazing. And that's what I'm most proud of, the teamwork that we've done to develop our practice, both as a clinical practice of excellence, but also a clinical trials center.
No, definitely. I mean, your practice is one of the leaders in the trials in the country. So thanks for everything you have done to bring new treatment options for our patients. And then the last question, Dante, I know you're never going to retire, but how do you want to be remembered once you retire?
I think like most people, I want to be remembered as a good guy. I want to be remembered by my family as being a good father, husband. I want to be remembered by my patients as being someone who cared and provided some value to their life by making their situation better. I want to be remembered by my staff as not being a jerk in the office and, being easy to work with. And I want to be remembered by the people that I've helped train along the way, either to go to medical school or during their fellowship, or just my staff who I've trained to learn how to become technicians and things over the years. So, yeah, I guess I want to be remembered as someone who contributed to the field, well, in a little way. I think each of us contributes a small amount, and all these small amounts really magnify in our field. We have great colleagues, and each of us is working hard to try to do our little bit to push the field a little bit further. And I think that I hope to remember that I played some small part in all that when it's all said and done.
And I remember you, Dante, as a fun person on the podium panels and as a good friend too. So I think you have achieved in your career, and it's always a pleasure working with you because you just make it fun, and you're super smart. And thanks for all the contributions you have made to the field and for your friendship. So thank you for being here, Dante, and participating in this podcast. We appreciate your time.
Arshad, thank you, and back at you at all those compliments. I feel the same way about you, and you make it fun and interesting, and I'm always impressed with your energy. I mean, you seem to be everywhere. I don't know how you do it. There may be two of you somewhere. I want to know where your twin is. I know there's another one of you somewhere. Yeah.
Thank you, and thanks for listening to this podcast.